Recording in progress.
So to introduce myself, so I'm Lizzie Harris. I'm a clinical geneticist. I work with the team mainly in facilitating diagnosis for patients with LGMDs. So I'm just going to talk about the importance of a genetic diagnosis. So start off by clarifying what we mean by a genetic diagnosis, and then expand a bit on why that matters. So without going into a lot of biology and details, I think we can probably all appreciate there's a difference between a diagnosis of something like asthma or a stroke or arthritis, where it's based on the kind of description of how that person is physically affected by their disease, compared to a genetic diagnosis where we're looking for a change really at the level of the DNA within the cell, a very precise change. So what is the gene that is affected, and then what is the exact genetic change that is affected? So when we talk about a genetic diagnosis, we mean finding the exact genetic change that is the underlying cause of that person's disorder. So this obviously involves genetic testing, and genetic testing has improved enormously over the last 10 years or so within a research setting, and now within the NHS as well. So previously we had to test genes one by one, just based on what we thought was most likely, looking at the person in front of us. And it could take years to get a diagnosis. But now we have something called genomic testing, and that really means just testing many genes all in one go. We're able to do that because there's been a big improvement in the technology, genetic testing technology we can use. So we do things called gene panels, where we look at a number of different genes. So in Newcastle we look at 80 different genes that can cause LGMDs and other similar conditions. That's our kind of first line test. And we can also do whole genome sequencing, where we look at all of a person's genes in one go to try and find the diagnosis. So initially, that sort of testing was only available in research, but we can now offer that within the NHS as part of the diagnostic testing service. So it's improved hugely. So the good news is that the majority of people with LGMD do get a genetic diagnosis and the benefits that come with that, but we know that there are an important minority of patients where we don't get that diagnosis, and that is still a very active area of research. So to understand what difference that diagnosis makes, I thought we should probably go back in time to before we had genomic testing. I wouldn't have put so many clicks in if I'd known. Another click. 2008, so 15 years ago. So this is before we had all of this genomic testing, even in a research setting. So this is a fictional patient, but probably is typical in some respects of many of the patients with LGMD that we see in Newcastle. So Anna is a 37-year-old woman who works in a gym, and she's got 2 children, a son who's 4, a daughter who's 8. And she's been previously fit and well, but over the last 4 years, so in her 30s, she found some things like climbing the stairs, getting up out of a chair, increasingly difficult, and it was impacting her ability to do what she needed to do at work. So she thought she would look into that. And she doesn't have any family history, we were always interested in that, but her son was a little bit late learning to walk, learning to jump. So she went to her GP, who was referred her to a neurologist who would have taken a history, examined her in a lot of detail, done some blood tests, including checking this protein in the blood called the CK, and arranged a muscle biopsy. And based on all of that assessment, she was told she most likely had a muscular dystrophy, but that this was a broad group of conditions. They were going to do some genetic testing to try to find out what the precise genetic diagnosis was. So because this was 15 years ago, that would have been 1 gene at a time. And at the end of that process, or perhaps after a number of years, we hadn't necessarily even come to the end of that process, but we were running out of genes that we thought could explain her presentation. We still hadn't identified the genetic diagnosis. So she was told she had a limb girdle muscular dystrophy, but we didn't know the exact underlying cause for that. So she kept patients like this who continue coming to our clinics every year. We still think about further genetic tests or other investigations to try to understand what's going on with them. They get their breathing checked regularly, annual heart checks, just because we wanted to cover all bases because we didn't know the exact underlying cause. So although a diagnosis of an LGMD is a sort of a clinical diagnosis, we would think, rather than a genetic diagnosis, and it's helpful for talking to other health professionals and for being clear about, conditions that might present in a similar way that it isn't, she still had a number of questions. So I think foremost in lots of people that we see's minds is, what does it mean for their family? Her son, with his sort of slightly late with some of his motor milestones, with learning to walk and things, she was always concerned, is this something that he has as well, and how is that going to affect him? And she has a brother and a sister, so thinking, is this something that they're going to have? No one else in the family seems to be affected, but I've been fine up until now, so what does it mean for my family? And also just the feeling that she hadn't met anyone who had exactly the same condition that she had, and she would like to know, are there other people out there, and how are they affected, how are they managing their lives, and what is the future going to hold for me? What's the people with my condition, how are they affected in 10 or 20 years' time? And although she kept going for these breathing and heart attacks, and they were always fine, but she had this concern every time, what's the chance really of finding something there, and how concerned do I need to be about that? Are there any other problems I might develop? And she would love to know, even though she understood that for, LGMDs there isn't a tablet you can take that's going to stop the disease, she would like to know whether people were doing research into that, is that something that people were looking into? So without that genetic diagnosis, those questions remain. So fast forward to today, 15 years later. So it's a slight change in the way that people with this sort of presentation with an LGMD would get investigated, because of the genomic testing being much more accessible and being so efficient, looking at so many genes in one go. So in this situation, she might not have a muscle biopsy straight away, because we can go ahead and do the genetic testing instead. So she would have had the panel of 80 genes that we do in Newcastle, looking at all of the LGMD genes and then some more as well. And in this situation, 2 changes were found in this gene called anoctamin 5. And it's worth saying that 15 years ago, anoctamin 5 wasn't even known to be a cause of LGMD. So this sort of reflects that our knowledge has improved. And so she's given this diagnosis of limb-girdle muscular dystrophy R12 anoctamin 5 related, which is a bit of a mouthful, and it used to previously be called LGMD 2L, but the LGMDs have undergone this change in the way that they're named to be a bit more systematic about them. So she has that precise genetic diagnosis. So just to go back to the questions that she had in her mind, so we can now tell her, what are the implications for her family. And this is a recessive condition, so the chance of her children being affected is very, very low. And we can also talk about what that means if she wanted to, for the rest of her family, conversations to have with her brother and sisters. And again, actually, this form of LGMD is one of the most common causes of LGMD in this part of the world, and so that actually means there is a whole community of people that she could get in touch with and find out about, and sort of get the benefit of that support. And again, we can tell her about the chance of having heart or breathing problems related to that. So in this condition, it's unlikely that those would be sort of significantly affected. So that is reassuring to her as well. And again, thinking about research, so we could tell her more precisely about what research is going on and natural history studies and that sort of thing that might be coming up. So I hope this fictional patient journey illustrates sort of the impact that having that diagnosis and that more precise information can make for patients. And this picture is in the Odyssey, because we know that patients who have a rare diagnosis, a rare disease like an LGMD, often can go on this long journey to find see the right specialist, get the right test done, and get their diagnosis. So I think a real sort of immediate impact of having a genetic diagnosis like this is that we can stop those investigations. So instead of coming to clinic and people umming and ahhing about, what is it, do we need to do something else, are we looking in the right direction, we can just stop the test and be certain that we've got the right diagnosis. And I think that has a psychological impact for patients. They can stop wondering. And I realise that I'm speaking to this to people who've gone through this journey, and I haven't. So you may have your own thoughts and things you'd like to say to me. So I'm sorry that it's not there. We don't have the chance to talk about that. So for the implications of the diagnosis for the wider family, so it depends on the inheritance pattern of the LGMD. So there are two different ways that limb-girdle muscular dystrophies are inherited. So like the diagnosis that our patient Anna has, most of them are recessive. So in that situation, brothers and sisters might be affected. And they, could be offered testing. Even if they don't have any symptoms, they might want to be tested to see whether they're going to develop it in the future, so they can plan, or they might not wish to. It's completely their decision. And we can be reassuring that provided your partner isn't someone in your wider family, or they don't also happen to have a family history of the same rare genetic condition, that the chance of children being affected is low. So that's really important information to be able to be accurate about for families. So less commonly, there are some rare forms of LGMD which are dominant, and in this situation you can get parents being affected. So the pictures at the bottom here is how we draw out family trees in genetics. So the squares are males and the circles are females, and if they're coloured in, it means they're affected. So on the right hand of the screen you can see that there's a father, a male, who's affected in the first generation, and then they've had children, and half of their children have it and half don't. So in these dominant conditions, then you can pass this on to your children. There's a 50-50 chance. Okay. And I think so. It's really important to be accurate in that information about the implications of the family for any future children, because people do make decisions about, a further pregnancy, having another child, and planning their lives based on this. And for some of so I've mentioned predictive testing, for example, in the recessive situation, like Anna's brothers and sisters, finding out whether they might have inherited the condition as well. For some of the conditions, there are options for testing in pregnancy in some situations, and also something called pre-implantation genetic diagnosis. So if there was a chance of having a child with the same condition, there's a possibility of using this IVF type technique where you can test the embryo in kind of lab setting and only have a pregnancy where the baby doesn't have that condition. So it's quite dependent on what the diagnosis is and the family situation, but those sorts of things are available, and obviously it's really important to base those decisions on really accurate information. So as well, a genetic diagnosis can change care and provide new opportunities for patients. So depending on the specific type of LGMD, there can be targeted screening, might be able to stop unnecessary screening. So if there's very little chance of breathing problems, if you're seen in Newcastle, you'd still get your breathing checked every appointment, because that's just what we do. But we would be less concerned about that. Same with heart checks. You may still get your heart checked, but it may be that is done with less frequency, and you can be just generally reassured about that. And just think about treatment in some other centres, perhaps, I don't think so much here, but people might be getting treated thinking they might have treating cause of their muscle problem, they might be given steroids or something like that. So if you have a diagnosis, you could be clear, actually you don't need to take those medications because they're not going to help, and they obviously have side effects as well. I'm sure other people today are going to talk about disease registries. So to join a disease registry, in general, you have to have a confirmed genetic diagnosis, so to be able to contribute to that collection of data and get the benefits of being sort of plugged into that kind of wider community, a diagnosis is really necessary. And similarly, clinical trials. So in natural history studies, I think some people here have been involved in natural history studies for LGMDs. It's really interesting to be able to look at how people's condition changes over time, and then look to see, is that they've all got the same underlying gene affected in their LGMD, but they've got different genetic changes. So does the particular genetic change affect the way that their disease progresses? And that's important because it might mean that if there are differences, we might slightly tailor somebody's care depending on their precise gene change. Or it might be that when clinical trials of a treatment are available, it might be that people with a specific gene change are selected or not suitable depending on what their information shows. So we know that for treatments of genetic diseases, sometimes if they're targeting the specific change in the gene, then some treatments will be likely to benefit somebody with one particular gene change and not benefit somebody else. So again, that's really important because we don't want to give people a treatment that's not going to benefit them, that may cause them harm with side effects, etc. So again, it all feeds into this. And as in the case with Anoctamin 5, actually, when that gene was first found, the role of that gene, the kind of protein that it was the recipe for, wasn't something that was understood to be important for muscles and how they work. So when that was found, there's new opportunities for basic research and understanding the underlying ways that muscular dystrophies develop. And of course, we're always thinking, are there opportunities for treatments based on that understanding? So in conclusion, we've got much better genetic testing, so it's really nice to stand here in 2023 and tell you about the good testing, because I think the NHS has done really well getting this rolled out. So more and more people with LGMD get a diagnosis, and that diagnosis is really the first step to providing the best possible care that we can for patients with LGMD and understanding what that means for their wider family, and ultimately working towards clinical studies and developing treatment.
Thank you.
We're not doing questions?
I mean, if there are any questions, we can take questions.
Could I ask a question? If people were diagnosed pre the genome becoming more advanced, do you think, is that something that would warrant the biopsies being revisited to see whether actually the original diagnosis is as accurate as it could be, or is that irrelevant?
So if they got a genetic diagnosis, say in 2015,
or say 08 or 06 or whatever.
Oh, sorry, yeah, 08.
Hypothetically speaking.
So we do sometimes revisit diagnosis, often if there's some sort of question in the family about a pregnancy or something. We want to make sure we're giving the right advice. But generally, I think if you were given a genetic diagnosis prior to that time, that we would generally think that it still stands. Yeah. So I think,
So I have a comment and a question. So one of the advantages, I think, of having a specific genetic diagnosis is to you can link with a community that's wider than just your local area. So there are online communities, certainly for my type, and I find that very helpful to be part of a worldwide community. But I wanted to ask about muscle biopsies specifically. So I had my diagnosis by biopsy 11 years ago, so I did well. But I'm hearing from some of my friends in the States that they think that they can get a full genetic diagnosis from a blood sample. Is that true?
Yeah, that's right.
So muscle biopsies aren't strictly necessary anymore?
No, I would say that. So generally what we do is go straight to genetic testing nowadays. And the muscle biopsy can be helpful because sometimes we find changes in a gene we're not sure the significance of. And then if we go back to the biopsy, that can help to inform our interpretation there.
So I presume there's a bit of me in the fridge at the hospital.
But
there are side effects to biopsy. I mean, it still hurts.
Right.
So
it is still necessary at times to have a biopsy, particularly if a diagnosis is not achieved. It used to be the gold standard. Now things have swapped around a little bit.
Easier to do it
go less invasive, but it may still be necessary to do it either because we haven't identified the gene, or Lizzie was saying that if we have identified the gene but are not sure that is the correct diagnosis, we need more evidence, then that muscle biopsy is still necessary.
Could it be possible for a child?
I don't actually know, other than LGMD, whether I've had anything. And this is the first time I've actually met anybody else with LGMD. So can I find out if there's been a blood test done?
Do you want to come and find me? I can take the details. That's fine.
Okay. Well, if there are any more questions, obviously we are around all day, and we'll be at the afternoon as well. So write down
Recording
your
stopped.
questions. And no question is stupid. Okay, so the next one is Robert, who's joining remotely. So we'll give an
Recording in
update on
progress.
physiotherapy and care management in British Muscular Dystrophy after this 3-2 drug trials.
Technology's complaining again.
We know, and you know best because you are the experts in your condition, that whilst within a drug trial or for the statisticians that we would like your change to be the same amount every year for everybody, that is not the reality. The reality is the condition takes all sorts of twists and turns. Sometimes things are stable, sometimes things are dropping off quickly, sometimes things are dropping off slowly. We need to, as a team of global clinicians that work with people with limb girdle, we need to all be speaking the same language. And so why we have natural history studies, both when we take your data in clinic and make you do lots and lots of things, including your breathing tests and asking you to step on the box or lift your hands up above your head, all of those things, is to try and understand and create a common language so that we as clinicians in Newcastle are all speaking the same language, but also all the other clinicians around the globe that look after patients with limb-girdle muscular dystrophy will all then be speaking the same language, so that when pharmaceutical companies come along and say we have a treatment option, that we can say as the clinicians, well, this is the test that should measure everybody with limb girdle, and this is a test about how much people change over time, and at this stage of your condition you should expect this much change, so that if there is a treatment, we need to be able to measure the treatment to see if the treatment halts progression or causes improvement and how it affects the condition. So we need to have that background understanding and that background common language around the globe of how we put you through your paces and measure you, so that we can then use that to understand, to make the diagnosis, to try and best predict and be able to predict when things are going to change, so that we can give you a bit more of a heads up. We don't have a crystal ball at the moment about when disease is going to change, but we would really like to. We're working on it. We want to know, if you come to clinic and you see me and you report what difficulties you have, and then I do some measurements, that I could then say to you, okay, this is a stable stage, or perhaps maybe walking is going to become more difficult over the next 12 to 18 months, so maybe we should think about looking at splints, maybe we need to look at walking aids, we need to think about your housing, your work environment. So by using those assessments are the building blocks to then try and give you the best management. And the natural history studies, as you know, your condition is very rare. It's wonderful to have more than 60 patients in the room today, but this is a big group of limb girdle patients. The condition is more rare than some of our other muscular dystrophies, and so in order to have lots of patients and lots of clinicians working together with you to do the assessments, we go global for our natural history studies. So I'll take you through a few of the natural history studies that are underway at the moment. Several of them are in Newcastle. We've got a long history of working in limb-girdle muscular dystrophy. The condition was first actually described here in Newcastle by two neurologists, Professor Walton and Dr. Natras, and that was in the 50s. They saw patients in the northeast with this pattern of muscle weakness that was affecting their shoulders and their hips, and they actually described the condition then. And then, as you've heard Lizzie speak, the genetics have been getting faster and faster, and the technology's been growing so that now we have more precise diagnostics. But that's, this is a long history in Newcastle of doing that. These natural history studies, some of them are global, some of them are single site. So some are just for one disease, and then more recently we've got global collaborations where we're looking at a number of different muscular dystrophies under one single natural history study. So for instance, there's several people in the room that have been coming to Newcastle over the last 10 years to participate in a dysferlinopathy, or limb-girdle 2B, or limb-girdle now R2, dysferlin related natural history study called COS, the Clinical Outcome Study for Dysferlinopathy. And we've been very fortunate to lead that study from Newcastle. It's been funded by an American charity, and that has brought us from assessing people with essentially the kitchen sink. We threw the kitchen sink because we didn't have a limb girdle specific test. in terms of your function. And so that study allowed us to put 200 patients together around the globe, 15 sites, put you through lots. Thankfully, you all agreed to come to Newcastle multiple times, upend your lives, take time off work, get assessed by physios and get really fatigued. But it has allowed us to actually develop an assessment now of your function for limb girdle that we now use in all the limb girdle muscular dystrophies, and is now used in more natural history studies around the globe. So that's the power of a natural history study. We went from having no common language for limb girdle in assessing your motor function, so standing, walking, lifting your arms up, to now actually having something that is commonly used in many clinical sites around the globe and in research.
Megan, is that the STAR test? Is that what we call a STAR test?
It's called the NSAD. It's the North Star Assessment for limb girdle type muscular dystrophies.
Could you say it again?
North Star Assessment for limb girdle type muscular dystrophies. Now, it's a really long name, and in clinic you may only know, you may not even notice it being done, because physio might be saying, right, are you able to lift up your head off the bed? Are you able to bend up your knee to your chest? Can you stand up off a chair? Can you lift your arm up to your shoulder? So it's a component of many parts, and we're probably just chatting through how things are going with you while surreptitiously scoring it for you in clinic with us. So we had, originally, early in 2004, we had a study with limb girdle 2A, cow pain, which was 4 sites. And then for the last, nearly 20 years now, in Iowa, Kathy Matthews has been leading a limb girdle 2I natural history study, and they have a big patient conference there and bring everybody together to do a lot of testing at the patient conference. We then had a study led out of Newcastle with us and Paris and London and Copenhagen, also looking at MRI and function in limb girdle 2I. Of course, COS has been going for the last 10 years. We've also, our colleagues at Nationwide Children's Hospital in Columbus, Ohio, have a broad natural history study of all limb girdle type muscular dystrophies. There's a single site, Vicky was describing Anno 5 or limb girdle R12 earlier, so there was a natural history study in Copenhagen just looking at that type of limb girdle. And then now we have these more global collaborations. The GRASS Consortium is a global study. There's about 17 different sites in it around the world that's looking at a number of limb girdle muscular dystrophies, but then we'll also look at other types of muscular dystrophies like Becker muscular dystrophy in the future. And again, it's using the COS model, bringing lots of clinical sites that are expert together to all measure patients the same way, so we can actually determine what we should be asking you to do and what would be a waste of time to ask you to do. What's irrelevant, because clinic doesn't go on for 3 hours, and we need to have what's most relevant to help manage your condition. Genathon have also a limb girdle 2I study, and Sarepta have recently started a study in sarcoglycanopathies, which is a more childhood onset type of limb girdle muscular dystrophy that Newcastle is participating in. So COS was our first big international natural history study, and of course that's now become the gold standard for what we need to do in other natural history studies around the globe. And we've looked at not just physio things, but biomarkers, which MRI, which muscles are changing, is there markers in the blood or the urine that might be useful to tell us about disease and how it changes over time. We've been looking at in the Genathon study for limb girdle 2I, and the baseline data has allowed us to see really nicely and actually quantify and document that in this graph, the NSAD scores on the vertical axis and people's lung function. When we get you to take a big deep breath in and blow it all out for as long as you can. That's the number that's on the bottom, on the x-axis. And what we can see is if you have limb girdle 2I or limb girdle R9, that there's quite a strong relationship. If your breathing's really good, generally you're on your feet and you're okay. As your breathing becomes more difficult, perhaps things like standing up from a chair and walking are becoming a lot more difficult. But by actually getting the data and publishing this sort of information, this will help clinicians around the globe be more aware of when and where problems might happen and how to manage. So yes, natural history studies, Thank you to everyone that is participating in them. Thank you for giving up your time. And the reason I hope the wider group will see is that the natural history studies don't just inform the study itself, but actually inform the wider care of limb-girdle muscular dystrophy. It helps our diagnosis. It raises awareness of clinicians around the world. You have a very rare condition. Some general neurology clinics may not see many people with limb-girdle muscular dystrophy. So by doing this type of work, it also has allowed us to develop some standards of care, starting in dysphilonopathy, but with wider plans, so that we can actually tell clinicians around the world positive experience on the table.
So now it's my turn. Over the next 20 minutes, I will try to go through the clinical trials that we are doing and also make a brief introduction of the clinical research that we are doing in the center. So my name is Jordi Diaz-Manera. I am professor of neuromuscular diseases here. I am a neurologist by training, so I am also a consultant and I see some of you in clinics. So as you have heard today, limb-girdles are a group of genetic diseases, and they are characterized because they lack a protein that is important for muscle function. So although there is not a specific treatment approved so far for muscular dystrophies, we do a lot of research in our center trying to find new cures, okay? So I think it's important to understand when we talk about treatments of limb-girdle muscular dystrophies, the different options that we have. And for that, I'm going to try to explain very briefly what is happening in your muscles when you have a muscular dystrophy. So we start from a healthy muscle that lacks a protein. This protein can be calpain, dysferlin, sarcoglycan. You have heard these names already. And this is producing a damage in the muscle. In the first step, there is a kind of inflammation in this muscle, and then we are going to start losing muscle fibers. And these muscle fibers are substituted by a tissue that is called fibrotic tissue or adipogenic tissue. It's fat and fibrosis. And this fat and fibrosis have not any capacity to contract. And therefore this leads to weakness and disability. Okay, so this is what is happening in the muscle. If we want to plan a treatment, there are different strategies. And the strategies that have been developed so far are trying to tackle all these different processes. For example, we can try to restore the protein that is missing, right? That could be a first option. And this is exactly what we try to do when we do gene therapy. So you have heard about gene therapy. And with gene therapy, what we try is to bring back a copy of the healthy gene. So you have a mutation, we want to put back a gene that is not mutated in your cells. And this way you will express the protein that is missing. There are many different strategies for doing that. We are going just to talk about one of them, which is, there's something that is here. Okay. One of them that is called gene delivery. So gene delivery is something that we do with virus. And what we need for that? Well, we need first a copy of the gene, and this is what we call the transgene. It's not something that we do in the lab. We create a gene that is very similar to the human version. Then we need a promoter, and this promoter is a small particle of DNA that ensure that your muscle fibers are going to express this gene. And then we need the transport, something that is going to transport this gene to the cells. And for that we use virus. It's incredible that after COVID we are talking about the virus that can be a cure. But if you think about it, it makes a lot of sense, because virus can arrive to all your cells very easily. So we take a virus, we put the gene inside, and we use it as something that will deliver the gene to your cells. And then...
Sorry, we can't see your slides remotely. I was trying to sort it. Sorry about this.
I'm sorry to hear that, Robert. So you have missed the most exciting part.
I was feeling that, but I was just trying to fix it, but I couldn't. Oh, sorry.
What about now?
Display settings, and you could duplicate. Display. Sorry about this, Jordan.
No, it's okay.
Can you do that to an adult or in their only thing?
Sorry, say again?
Can you do that to an adult or
yes.
They have to happen in an embryo?
Yes, we can do it to an adult.
Do we need to win the lottery?
No.
Do we need to win
the temperature little less better?
It is better now.
Okay. So I'm sorry, Robert, but we are having technical problems with that. So I will try to continue. So this is what we need, and just to show you an example, this is what is happening with we inject the virus. We inject the virus, you can see there on the screen, and you can see that the virus will arrive to the cell, will deliver the copy of our gene that will go inside, and the cell will start producing the protein that is missing. Okay, so, well, all this story starts always doing preclinical research. And today I will go through that because I want to show you the importance of the preclinical research. So, in limb-girdle, there have been some studies doing gene therapy in animal models. So we use different animal models that mimic more or less the phenotype of patients that have the same problems, and we try to treat these models with these strategies. So here, for example, you can see that the first thing that we need to demonstrate is that when you inject the virus arrives to the cell starts producing this gene and restores the expression of the protein. And this is something that has demonstrated in murine models of limb-girdle muscular dystrophy. What's happening now? And then we need to demonstrate that this protein is functioning. It's not only there, it's also functioning. And this is something that has been also demonstrated in these animal models, that there is an improvement in the tissue structure, so the tissue looks better, there is less fibrosis, there is less inflammation, and also there is an improvement in muscle function. And this has also been demonstrated in the animals. So we are closing the cycle. So we can put a treatment that restores the protein, this protein is functional, is improving your tissue, is improving your function. So now we can go to clinical trials with patients. And what is the experience so far? In limb-girdle, we are talking about limb-girdle muscular dystrophy. We always start with local delivery. So these are 15 human trials. So it's a new medication. We just want to inject this in a muscle. And this has been done doing an injection in this small muscle in the feet of the patients, just locally, to see what happens. And the first results are showing, as you can see, that there is a restored protein expression, so the protein is there again. And there is a bit of inflammation locally and also systemically. And you will see later that this is one of the problems that we can have with gene therapy, that sometimes as we are expressing a protein that is new, there can be a bit of inflammation. It's something that we will need to control. But of course, we don't want to treat just one muscle. We want to treat all muscles. And for that, we need a systemic delivery. So an intravenous injection. And there is already one trial in patients with limb-girdle muscular dystrophy produced by beta-sarcoglycanopathy, which is one type of limb-girdle, where patients have been treated intravenously. So these are some of the results. There have been 3 patients that have received a low dose of this treatment and 3 patients that have received a higher dose. And as you can see here, the patients were, of course, with a diagnosis that was confirmed. Genetically, they were able to walk 100 meters in the test, and they were treated with steroids in order to reduce a bit this inflammation. And what happens to these patients? Well, the first result that we have from the company that is called Sarepta showed that the patients were able to restore the protein, okay, in the muscle to an extent that is between 30 and 60% in a high number of fibers, more or less between 50 and 70% of the muscle fibers. And this has led to an improvement in their muscle function using this scale that Meredith has just presented, the North Star, the NSA. So the results are just 1 year of follow-up. We are talking about just a few number of patients, 6 patients. We need to do more, but this is the first step of a very exciting story. That is that we are for the first time treating patients with limb-girdle muscular dystrophy with gene therapy. And we are about what is called a phase 2 clinical trial with a larger number of patients with this condition to treating with gene therapy. And if we succeed, this will lead to many other trials because there are plenty of them that are in development. So what are these other trials? You can see here a list. There is a trial for Limb-Girdle muscular dystrophy at 9, which is the 2I. There is a trial for calpain. There is a trial for dysferlin. There is a trial in planning for ANO 5. So the good thing is that this strategy can be applied to many different Limb-Girdle muscular dystrophies. Because we have the tool that we need, we have the transport, we just need to change the gene. And then, why is it important that we succeed with one of these diseases? It's important because we impact all the others. If we can demonstrate that we can benefit patients with beta-sarcoglycanopathy, this means that this treatment could work in all other types of muscular dystrophy. That's the reason why, if you have, for example, a calpain, and you say, oh, there is no treatment for me, probably you are right. And now, we don't have a treatment for you. But there is a treatment for another type of limb-girdle that sooner or later will impact the development of a therapy for you. Because if we succeed now, we could expand this treatment. If we fail now, then the whole program is at risk. That's the importance of these first clinical trials. Okay, I know that I'm running out of time, and I just want to go back to this graphic. So you can see here that there are other things that we can do. So for many years, there have been other treatments trying to tackle inflammation, for example, using different anti-inflammatory drugs. Steroids are given to patients with Duchenne, and there are other anti-inflammatories that have been used in patients with muscular dystrophy. So some of the strategies try to reduce the inflammation. You can see here reduction in fibrosis and fat expansion. This is another strategy, and I will talk about that. So there are new anti-fibrotic treatments that try to reduce the fibrosis in the muscles that are also being developed. And you can see here, the last one is increasing the muscle tissue. So once we have fat and fibrosis, what can we do? This is what we call cell therapy. And probably it rings you a bell, all these stem cells that for many years we tried to develop. So it's true that the results were not the best, and this is a strategy that needs to be reconsidered. But there are many groups that are working on that as well. So I'm going to pass these slides because I'm conscious of time, and I'm going to move to the next presentation that is about the basic research that we do, and the links with this, and I don't know what is it. Heather?
Is it in the folder, Heather?
Okay, so maybe what we can do is, because there is this problem with the slides, we can stop here. We'll do a coffee. I don't know, you have questions, maybe you have questions, and we can start
recording stopped
with the next presentation. Any questions?
You know, you're doing all these studies. Have you put it down any technical general population?
That's a good question. So, of course, we first need to do the trial. And if they succeed, then they need to go through approval from the pharma organization that needs to take. Almost takes time. We're talking about 3 years. Even if we demonstrate that it's it's always 3 years until this can be available.
So for
But this segment of dystrophy 2I, which is R9, and this is the largest community patient with muscular dystrophy in the Northeast. That is about to start. Actually, Volker, you mentioned that you are going to infuse a patient in the next weeks, right? So Volker now is the PI of that study. So you have requested, you can also reach to him later. I think it's a good announcement for everybody to know that the trials are arriving, the trials are here. So in the next 15 minutes, I would like to comment as well in the importance of the preclinical research. So I am also very interested in what's happening now. So I'm also very interested in preclinical research, and I think this is a good opportunity to share with you what we are doing in the center. So what is preclinical research? So preclinical research is the research that we do in the lab, and it's the research that we do trying to identify the causes of the diseases, of trying to identify biomarkers. And we work with muscle samples, we work with cells, we work with animal models, and we work more and more with bioinformatic analysis. We are using, implementing computing science into clinical, into research. So why is important to do preclinical research? For many reasons. But one reason is for having a better understanding of the disease mechanisms. So if we don't understand what is going on with a disease, we will never find the cure. So that's one of the important points. To test new therapies, of course, if we want to develop therapies, we need to test them first in models of the disease, and also to identify new biomarkers. You have heard before that there are functional biomarkers, scales, that can be used. But there are other types of biomarkers, molecules that can be expressed in the blood that can be also useful to diagnose or follow up patients. So what do we do here? We do what we call translational, patient-centered preclinical research. So this means that everything starts with the patient. Okay? So we are doing clinics, we are listening to the patients to identify what are the main problems that patients develop. This can be sometimes fatigue, weakness, stiffness. We want to know how the disease progresses, what are the organs that are involved, for example, and if there is any treatment available for the disease. The next step when you do translational research is to meet with the people, with the team that is doing research, and discuss what we have heard from the patients. Describe the observations performed, generate an hypothesis. For example, we want to look, for example, at the heart, because most of the patients are complaining about cardiac problems. We then review the scientific literature because we don't want to repeat what others are doing. We want to do something new. And then we plan the experiments. The third step is to do the experiments. So the experiments are done, of course, in a lab setting. When we are talking about preclinical research, we do analyze the results, we review the results, and we generate solutions. And that's the most important part of translational research. We go back to the clinics because we want to bring back to the patients what we have discovered in the lab. And this can be treatments, can be solutions, can be new diagnostic markers, trying to improve the quality of the care that we deliver and also the quality of the life of our patients. So that's the cycle of the translational research, that starts with the patients, go to the lab, and go back to the patients again. That's the main aim. So, for doing translational research, what do we need? We need patients, of course. That's the most important part because everything starts with the patients. We need samples. And you will hear during the next 10 minutes that I insist in the importance of samples. That's the reason why when you come to the clinics, we always ask you if we can have a serum sample, a plasma sample, if we can have a muscle biopsy, because we need to do research. And this is based in the sample that we obtain from patients. If we don't have samples, we don't have anything. We need a lab. We have a lab. And important, we need researchers. So we need funding to pay the salaries of the researchers that are going to do the research. And that's the reason why I put there, we need money. Without money, there is no research. That's for sure. So, just an example of the translational research that we do here. So, one of could be an example. So, we are very interested in our lab in studying the fibrotic process of the muscle, the adipogenic process of the muscle. So, how the muscle becomes fibrotic and adipogenic. And here I'm showing you a study that we have recently finished where we have taken muscle biopsies of patients with a muscular dystrophy at different stages of the progression, so from mild, moderate, or advanced. And we have used a new technology that allows us to study these muscle samples. And we have been able to characterize that when the disease progresses, there is an increase in fibrosis, there is an increase in this tissue that is substituting the muscle fibers, as you can see, and in this red thing that you can see here on the bottom at the left. And we have observed that there is an increase in fibrosis associated with an increase in inflammation and with an increase of cells that are called fibrotic progenitor cells. So we want to use these cells as a model for research. So thanks to the muscle biopsies that we have stored in the biobank, we have developed a protocol that allows us to thaw the muscle biopsies, put them in culture, the cells grow from these muscles. we can isolate these cells, we can immortalize these cells, so these cells can grow forever in the lab. And thanks to a protocol that Esther has developed, Esther is going to be here this afternoon, we can differentiate these cells into fibrotic cells and adipose cells. So now we can start looking for treatments with these cells that come from the patient that we see in clinics. Okay, so this is what we are doing in the lab. We have identified a large number of treatments that can reduce the differentiation of these cells into fibrotic cells. And we have now 2 PhD students, you have them here, so and Priyanka, that are trying to characterize these treatments and moving them into animal models. So this is something that we have done in the past already. We have experience working with these animal models. Esther and Patricia here in the slide have been working for many years on that. And we have treated the animal models with these antifibrotic treatments, demonstrated that the mice are better. And you can see this red tissue in the center are the animals not treated. And on your right, you can see that with the treatments, we reduce fibrosis. So this is an example of what we do. But we want to also develop new models for research. And we are very interested in creating what we call muscle organoids. So from cells of patients, we can create an artificial muscle in the lab that you can put in a dish and can contract. And we can create these molds that you can see here, the plastic things. We put the cells there. We have these muscles that are three-dimensional. And we can treat them and see how they contract. And this comes all from the cells of the patients that are in the muscle biopsy. So I think that there has been a question before. Is muscle biopsy important? Okay, for the diagnostic process, probably now we have better solutions. But if we don't have muscle samples, none of what I have shown you would be possible. So that's important because samples are not only for diagnosis, are also important for research. And going back to this, that's exactly what I want to mention. So here in the center we have a large biobank that is managed by Dan Cox and by Andrew, and we are collecting all the samples from research that comes from the clinics, serum, plasma, urine, muscle, skin, go to this biobank that is you helping us, but also many centers around the world to do research in rare neuromuscular. Just some pictures of people that you don't know because they are not in clinics, but they are in the lab. They are doing research in improving the diagnosis and also improving the identification of new therapies. And I'm going to stop here just to show and thank to all the team and the funders, and of course the whole John Walton Muscular Dystrophy. This is my email and my Twitter account, and I'm happy to take any questions that you may have. Questions from anyone?
So I'm in the Genethon natural history study. Last time I was here, they were starting trials, I think. I was in, I was out, I was in.
Okay, I think that's a good question for yourself.
And I think it was down to initial findings on inflammation of the heart, and therefore I'm below 50%, so I was kind of screwed at that point for myself. Is Genethon now?
It's weird with something like deep massage fit with trying to tackle fibrosis.
That's a good question. We don't know if there is any mechanical way outside like a massage or other techniques to reduce. We don't think that that's going to have a great impact in patients, so we don't think that that's something that could help too much. But of course, it could help in other things, like like stretching, reducing contractors, etc. But probably not too much in fibrosis.
Okay, so it wouldn't necessarily reduce the fibrosis. Would it mean stop it developing or no?
Not really. I don't think that the massages are going to be a therapy for this disease. But I think it's something that can some of the symptoms that you may have, like being stiff, having pain, that could be handled, but not in terms of the progression of the disease.
Right. Okay. Thank you.
Just again, probably a selfish question, but just wondering how can people avoid, like, for example, they wanted to be in trial, like, is there anything that they can avoid to be exposed to these kind of virus?
You mean what you need to do if you want to be in a trial, for example?
Like for example,
Can you avoid anything like if you want to take part in a gene therapy trial? I guess this is your question. Can you avoid being exposed or
to the virus?
No, I don't think that you need to be worried about that. I think that you need to do normal life, and of course, it's our work to develop solutions to overcome some of the limitations of gene therapy.
Because for example, my situation is I do get exposed to people who has different problems, like different viruses. So then I go back and I don't want to give it to my daughter, if it makes sense. So it's just like those kind of things, so we can't really do anything.
No, because the reality is that viruses are everywhere, and not only with the people that you can work with or meet with, they can be also with your relatives. And we cannot go into a kind of hysteric situation. We try just to avoid any social contact, just in case I got infected for a future treatment.
Sure.
So there are now new also strategies trying to treat patients, pre-treat patients with anti-inflammatory drugs, with immune suppressive drugs to allow them to participate in the trials if they have high antibodies. This is something that is new, needs to be tested, but of course this could be one of the solutions for the future. So I think that my recommendation would be you need to live a normal life, and then let's see what it comes. And if there are these problems, we researchers, we need to find a solution, and not you avoiding having social contact. Okay?
And probably this, I don't know again, but it is relevant.
I'm Melissa.